In the early 2000s, other companies were starting to catch up with us in terms of sensitivity. The gold standard had kind of moved away from the API 3 to the API 300, the 3000, the 4000. The 4000 was great, but the ability for us to offer a model of every one of the triple quadrupoles with Q-trap functionality gave us a huge, important differentiating feature that our competitors did not have.
SCIEX has traditionally been the leader in triple quad technologies. We're the first ones to bring the LC-MS to the mass market. So the challenge is continuing to innovate, making sure that we have the next best thing that will enable our customers to make the discoveries that they need to do in order to progress their research or help make people's lives better. So we're always looking to find the next greatest thing for our customers to make them successful in their jobs.
Every biological system is incredibly complex, more complex than we can even imagine. The business of taking a large cohort of patient samples and getting relative quantification has always been extremely difficult. I think a game-changing technology like SWOT-MS had to come along.
So SWOT acquisition came about from understanding that customers wanted to quantify many more molecules than realistically they could build mass spec methods for. The studies that biologists want to do are not just 3, 4 samples, 5 samples, they're getting to the hundreds and thousands of samples. And from there we had to come up with strategies on how to process that data fast enough and enable the end user to take those numbers and turn them into something that they really understood.
The Stoller Biomarker Discovery Center was developed in order to find biomarkers of, say, risk of having a certain cancer. We need to have signifiers, biomarkers of response to therapy or diagnosis.
SWOT is our preferred approach for many reasons. It has high reproducibility, high sensitivity, very high dynamic range, and the most important thing, we can get very efficient and good proteomic signature maps in almost a couple of hours.
I think the speed and sensitivity of the instruments has increased a great deal. This generates a lot of work in terms of informatic analysis, but it also generates a lot of opportunities as well.
For PhD students and also for research technicians that worked at the Stoller Center, I can see a great future in proteomics.
We've had quite a lot of different diseases. We've had Alzheimer's, we've made some good progress with that. We've had ovarian cancer. I've worked with some heart disease. And mainly we're looking for a variety of different biomarkers, but they can tell us a lot of different things. So really that's the main bent of the study, is to build up these almost like fingerprints for each person and for each disease so that we can predict how people are going to respond to illnesses, what diseases they're going to get, that kind of thing. That's the main focus of the research.
You could have a healthy diet for all your life and you're still prone to cancer. That's still misunderstood. It's not well known. The technology we can develop helps to understand at the submolecular level what is actually going on.
There are more drugs coming out and faster. The drugs are better, and ultimately, if we have that scenario, people are going to get the drugs and we're going to save lives.
So the technique SWOT allows quantitation of metabolites, proteins, lipids. It allows us to understand what actually is a snapshot of a body. And if you put that together in a longitudinal time, you can understand how the body changes. If we can detect cancers early, then in reality, you won't lose your parents early. You won't lose your children. And we might be able to actually make it a better place because of that.